Hi everyone, my name is Angelina and I'm a fourth year PhD student at Oregon State University. My lab is the Henkel Lab, Sarah Henkel's lab, and I am currently at the Hatfield Marine Science Center, which is where our lab is located. And my research is really focused around kelp physiology. We have a lot of kelp on the coast of Oregon. It's a primary producer. And it's a foundation species for our intertidal and subtidal systems in the ocean. And I'm focused on their physiology, so how climate change affects their physiology, such as like their photosynthetic rates and their respiration rates. And I'm also interested in how they impact the communities underneath them. So I've been doing a lot of field work and studying their communities and what happens when the kelps are lost or when the kelp canopy changes. How does that like cascade down to the rest of the community? And then I'm also interested in just like what happens to their physiology as climate is changing or just as oceanographic conditions change. So today I'm going to walk you through how I look at nutrition of kelp and how their nutritional values change and what I use in the lab to quantify that. So stay tuned. Okay, so to introduce you to the Henkel Lab, this is the door, this is where you walk in. This lab is pretty heavy boat work, so a lot of my other lab members like to go out on boats. So we have a lot of equipment. This is a CTD, which is a big device that you drop off the side, the back of the boat, and it will track basically like all oceanographic parameters for you while it's underwater. And then we have some cables to look at how energy for wind energy lines impacts studies. We have a ton of buoys, and then we have a bunch of field gear, and then everything else in this lab is samples. And these are all samples that have come off of the boats. So most of these, like recently in the last couple of years, they've been collecting in an absurd amount of sand dollars. So these are just like hundreds and hundreds and hundreds of samples of sand dollars that need to get sorted, and the species need to be identified. And one of my kelp samples, so this is Chaetophyllum sessile. This is a frozen version, so that's why it's all stiff. But it is an intertidal species, and it is a foundation species. It's commonly referred to as the sea cabbage. It has a very cabbagey shape when it's sitting upright in the field. And then basically what I do with these samples, these are for nutritional analysis. So I'm gonna take this individual and try to figure out how many calories of energy are kept within it. So this is what it looks like to start with after it's been frozen. And then I will pop them into the oven so they dry out, and then they get ground up, and that's what comes out into like a nice little dust like this. And so a bomb calorimeter is a great way to check energy and how much energy can come out of a sample, an organic sample. So this is what it looks like. This is the dried kelp right here. So you can see I've weighed out exactly 1 gram of it. I've put it into this nice little chamber, and then it's got an igniting wire right here. And then this whole thing will get put into the rest of the chamber and encapsulated. And then I will use this oxygen tank to pressurize this canister up to 35 atmospheres, so much higher pressure than just normally outside. And then this whole setup will get put into this nice little water bath, and then some igniter cables will get added to the top of this lid right here. And so what happens is that the sample, I can hit this nice little button right here, I can ignite the wire then will burn, and it will drop down, and it will ignite the kelp. And then when the kelp burns, that releases energy right when things burn, and then that heats up the water. And so I basically track that temperature change of what's happening with the water, and then on my little sheet I can calculate how much energy is coming out of the sample as it burns. The main question is, how does the nutritional value of kelps change seasonally? So I've been tracking them every season for the last 2 years. And also, how do they change in response to like oceanographic conditions? So for example, we had a marine heat wave off of the coast of Oregon and Washington in the middle of 2023. So I was able to capture if there was any associated changes with that heat wave. So far I haven't seen any. like changes in relation to heat waves or oceanographic conditions. The species that I work with seem to be relatively stable in their caloric content regardless of what's happening, which is pretty good because they are foundation species, so we do want them to be stable and keep producing habitat for everybody else. But yeah, so that's one of the things that I'm doing for my PhD.